Robin completed his PhD at the University of Michigan this year in April under supervision of Dr. Yang Zhang. Prior that to joining this PhD program, he completed his master degree in biochemical engineering and biological chemistry at the University of Michigan. And his research involved development in deep learning methods for protein. There was this one method, protein design, and also for RNA structure prediction. And I hope we can also learn about his future plans. Is it postdoc in science or in academia? So we have almost 1 hour to discuss. And I think if this is okay with Robin, we can stop at any time and discuss the things before we get to the end. I think this usually works pretty well.
Yeah, absolutely. If you have any questions, feel free to interrupt me at any time. So
okay, so the screen is yours, so you can go full mode, and I will keep admitting people.
Okay.
And if you have any questions, then just write in the chat or raise your hand. I will keep looking around.
Okay, great. Yeah. So thank you so much for the introduction. So today I'm going to be talking about our work in RNA tertiary structure prediction using potentials from deep learning. And essentially, the motivation behind the work is similar to what we see in protein structure prediction, probably starting back in 2017 with methods like RaptorX, and then they were later extended by L-Fold, the first version of it. And then probably even more recently, methods like RosettaFold, where they're based on the distance map-based folding. So what's the distance map-based folding? Probably most people are familiar with, but the goal of these approaches is to essentially simplify the problem of the RNA 3D structure prediction, because it tends to be a little bit tricky, and it's a little finicky to train it, at least from my experience, for the end-to-end. But instead of directly training it to predict the 3D structures directly, instead we can look at some simplified view of the structure, such as the distance maps. So, for example, if we consider all the distances between your C4 prime atoms or your N1, N9 atoms, then you can get a pretty clear overview of the topology of your RNA of interest. And in DeepFoldRNA, we consider 3 distance restraints. So, namely, they're the N1, N9 distance restraints, which is the first atom of the nitrogenous atom of the base. And then we have the backbone C4 prime distances, as well as the backbone phosphate backbone distances. And then on top of that, probably starting with methods like TR Rosetta popularized the orientation prediction in a deep learning framework for proteins. And these methods have since been extended by some recent works in RNA structure prediction, where instead of just predicting the pairwise distances, we can also predict the 4 atom orientations between the different nucleotides. So in DeepFoldRNA, we consider 2 different orientations, and they're the orientations formed by the P C4 prime and N atoms from residue I, and then the N atom from residue J. And then likewise, since this is an asymmetric restraint, we also have to consider the P C4 prime and N atoms from residue J and the N atom from residue I. And we call this the theta orientation. And then finally, we have our symmetric omega orientation, which is formed by the C4 prime and N atoms from residue I, and the N and C4 prime atoms from residue J. And it turns out that the orientation information is actually pretty important. So we know that from work in statistical energy functions, that those methods that tend to be based purely on the distance potentials generally don't perform as well as those that also include the orientation information, especially for maybe more complex folds in proteins like beta barrel folds that have more complicated hydrogen bonding interactions. And so here's the network. Obviously, so we know what we want to predict is we want to predict these distance and orientation restraints and hopefully be able to use them in some sort of useful way to generate our 3D structures for our RNA of interest. And the way that we do that is we take our query amino acid or nucleic acid sequence, and we search it through our sequence databases, which include databases like the RNA Central and NT databases. And from that, we can obtain a multiple sequence alignments of all of your homologous sequences. And what this multiple sequence alignment represents is all of the evolutionary information for a given RNA sequence family. So we know from works in direct coupling analysis and the earlier covariation-based methods that we can, that the multiple sequence alignments can essentially encode a lot of the position information by looking at covariant residue pairs. And then on top of that, we use the predicted secondary structure and the paired sequence information. And so this embedding represents the pairwise geometric relationships between each position. So essentially what we have in our network is we have, after we embed our multiple sequence alignments, and then we embed our paired predicted secondary structure and our paired sequence, we have two major embeddings, which is the MSA embedding and the pair embedding. And like I mentioned before, this MSA embedding captures all of the evolutionary information from your homologous RNA sequences. And then the pair embedding captures those pairwise distance and orientation relationships. And the basic architecture of the network is similar to methods like introduced by AlphaFold 2, the evoformer methods, where we first take our MSA embedding and we process each of the rows in our MSA to extract our positional information. And then in order to ensure consistency between our MSA and pair embeddings, we add a bias to the row attention procedure for the MSA embedding. And then the second way we process our MSA embedding is through column attention to extract which position or which sequences are at a given position are the most informative to your overall goal of structure prediction. And then we can take the outer product mean of our MSA embedding to update our pair embedding. And then we can finally process our pair embedding using triangular self-attention that was introduced by AlphaFold 2 as well. So it's sort of the idea here is to build upon what we've seen that has been successful in protein structure prediction, and whether or not that these networks that were trained on that have demonstrated pretty remarkable success in proteins, if they can do the same for the RNA. And so then at the end of that, we have our final pair embedding that we pass through our 48 transformer layers. And we can just do a pretty simple linear projection to get our predicted distance maps for each of our three distance features, which are N1, N9 distances, and our C4 prime and P distances, as well as our two inter-residue orientation maps. And so here, just a quick note, because we divide our distance maps from 2 to 40 angstroms. So we generate bin predictions for the distances. And then we also have one bin that corresponds to whether the predicted distances between two atoms should be less than 2 angstroms, and a bin for whether or not that distance should be greater than 40 angstroms. And the orientation maps are divided from 0 to 360 degrees into 24 bins. And then we have one bin for no interaction. And no interaction here just means that the distances between the two residues is greater than 40 angstroms. So we define that as the cutoff point for there being an interaction. And then just as a final word for the inputs features, we take the MSAs generated by RMSA, which I'll explain on the next slide. And then we use the PETFold reliability scores as the predicted secondary structure representation, which we can generate directly from the multiple sequence alignments. So here's the pipeline for RMSA, which was developed by Chenxing Zheng, who was a former PhD student in the lab. And essentially, the overall goal of the pipeline is to iteratively build up a progressively deeper MSA. So in total, there are 5 different stages. So in the first stage, we take our query sequence and we use BLAST-N to search it through the RNA Central and NT databases. And then we can pick up a significant number of BLAST-N hits. And then we perform we realign those hits using NHammer. And then we use the predicted secondary structure by RNA fold, as well as the alignments from NHammer to build our CM model, our covariance model here. And then we can research that covariance model through our BLAST-N hits to get our stage 1 MSA. And so then if the MSA from stage 1 is sufficiently deep, meaning the NEF value is greater than the cutoff NEF value, which here is 128, then we stop the procedure and we take this stage 1 MSA as our final multiple sequence alignment. If that's not the case, then we go on to stage 2, where we take our covariance model 1 from the previous step, and we search that using CM search through the RNA Central database to get our stage 2 raw hits. And on top of the hits that we pick up from RNA Central, we also add the BLAST-N hits from the previous round to this pool of raw hits. And then we use CM search to realign those and to get our stage 2 MSA. And then stage 3 is very similar. The major difference here is that instead of using CM search to search the RNA Central database, we use CM search to search the NT database. And similar to before, we take our stage 3 raw hits, which include the hits from the NT database, as well as the hits from stage 2 and stage 1. And then we can perform CM search realignment to get our stage 3 MSA. And so the final 2 stages are a little bit different than the previous 3 stages, which is that we take our query sequence. So we're no longer starting from our covariance model from the previous steps. And we research our query sequence through the RNA Central database using BLAST-N to get a new BLAST-N alignments. And then we can add our predicted secondary structure by RNA fold, and as well as build our covariance model using CM build to get our CM2, our covariance model number 2. And then we search that covariance model through all of the previous hits that we've detected. So from stage 1, 2, and 3, we search the covariance model through those pool of hits to pick up our stage 4 MSA. And then stage 5 is quite similar to stage 4. The major difference here is that instead of searching the query sequence through the RNA Central database, we search it through the NT database. So overall, the goal here is just to generate hopefully progressively more sequence hits. So of course, if we're able to generate enough hits in stage 1, then we don't have to move on to the next stage. And likewise, if we're able to generate enough hits in stage 2, we don't have to move on to the next stage. So it's really just helpful when you have very few homologous sequences and you want to build up a reasonable multiple sequence alignments. Okay. And so finally, for the network here, so in addition to the MSA-based network, we have a single sequence network. So it turns out for the RNA structure prediction, actually the single sequence networks seem to be much more reasonable than what we see for proteins. And in this case, the pipeline is essentially identical to the network that's based on the multiple sequence alignments. The major difference here is that we don't have our column attention because we don't have multiple sequences. But other than that, the features and the pipeline are all pretty much identical, where we take our query sequence and we embed it into our network to get our sequence embedding, and then we process that using our multi-head self-attention, where we add a bias from our pair embedding to ensure consistency between the two different embeddings. And then finally, we can update our pair embedding based on our sequence embedding using the outer product. And then finally, we process our pair embedding using the triangular self-attention scheme. And at the end of that, we get our predicted distance and orientation maps. So really the benefit of having these different multiple networks is that we're able to generate an ensemble of different restraints. So it's pretty helpful when, for example, one method or one network may generate unreasonable predictions, but if the second network has some complementary information, it could help offset that. And we see that typically when you ensemble multiple different networks, that the performance is greater than any of the single networks themselves. And here, the major difference in the secondary structure is that it's based on the RNA fold reliability scores as opposed to PEG fold, which uses MSAs. But other than that, all the features should be similar except for the MSA. Okay, so we collected a training data set of about just under 3,000 RNA chains, single chains, from the PDB that was collected on February 7th of 2022. And we didn't set any maximum length or resolution cutoff because, as probably everybody is aware, that there is very limited training data for RNA. And especially at the fold level, you may have a number of sequences, but they all share the same fold. So there are a lot of well-represented folds like tRNA, where you can, of course, generate many training examples. So I think it's a huge limitation for the RNA structure prediction, as opposed to the protein structure prediction, where we have a good majority of the folds that we would expect in nature are represented in the PDB library. Of course, we can always find some unknown folds, but I think it's certainly far more complete than what we have for the RNA structures. And then just to help enrich the data here, we take a distillation set of 16,842 RNA structures from the BP-RNA 1M database, which is a database that has information on the predicted secondary structures from the sequence covariance models. And what we do is that we first train the network based on this PDB data set, and then we generate labels based on our network that was trained on the PDB data set for our distillation sets. So then we can use those labels in our second round of training as sort of the ground truth, even though it may not be necessarily a correct prediction, but even then, it seems like the results for the distillation training are typically much better, which is, I guess, an interesting phenomenon of machine learning. So, and then finally, so once we have our distance, our native structures, in our training set, we can generate the labels by taking the distance and orientation maps calculated from the native structures, and then we discretize them into their bin values, similar to the predicted distance and orientation maps, where for the distances, we have 38 bins from 2 to 40 angstroms, and then a bin for less than 2 angstroms, and a bin for distances that are greater than 40 angstroms. And then for orientations, we have 24 bins from 0 to 360 degrees, and 1 bin if the distance for the residue pair is greater than 40 angstroms, meaning that 2 positions don't interact. So I'll just pretty quickly cover this. So obviously, since we had the binned predictions, we can train the network using the negative log likelihood loss between our predicted distance distributions and our native distance distributions. And on top of that, we also add a BERT style loss where we randomly mask about 15% of the nucleotide positions, and we ask the network to predict the masked positions. And this is another way that we can utilize unlabeled data in our training procedure to try to augment the procedure, especially given the limited amount of RNA structures we have. And when we did the training, we would take random subsamples of the MSA. We would, where we'd select up to 128 sequences for each training sample. And then we would perform random cropping to augment the data that we have. So if a RNA is greater than 256 nucleotides long, we can generate multiple training examples from a single RNA. And then lastly, the distillation set we incorporated after the initial training on the PDB data sets for about 90,000 steps. We trained it on the PDB data set, and then for about additional 90,000 batches, we trained it on the combined PDB and distillation set. Okay, so the last thing I'll cover in the methods is the folding simulations. So obviously, to have the predicted distance and orientation maps is good, but what we actually want is the predicted 3D structure. And it's fairly simple to get the predicted 3D structures based on the distance and orientation maps. So what we can do is that we can take the negative log likelihood potential of our predicted distance and orientation distributions, and we can use that as the potential for very rapid gradient-based folding simulation. So we use L-BFGS minimization, and this has been shown to be pretty robust for the, at least for the protein structure prediction. And I think it's fairly reasonable also for the RNA structure prediction, given a sufficient number of predicted distances. So the way that we calculate the potential here is that essentially we take, sorry, my screen went back. So essentially what we do is the output of our network will be the probability distribution for each distance bin, and then you'll have a distribution that's above whatever cutoff you have. So in our case, 40 angstroms. And what we do is that instead of just taking the negative log of this probability distribution, what we try to do is we try to normalize that by the amount that's in our final distance bin. And so what this allows us to do is that, for example, when you have very little of your distribution outside of that cutoff, then this is a pretty confident prediction here, and your curve will be approximately Gaussian. But in the case of, for example, you have about a medium sum or somewhat more significant amount of your distribution outside of that cutoff, instead of introducing weird artifacts in your energy function by just taking the negative log likelihood of the probability distribution, if you normalize it by the probability from the final cutoff bin, then you're able to get a much smoother potential. And then lastly is the lowest confident case, where the majority of your distribution falls outside of that cutoff range. And in this case, essentially, you're still able to generate a smooth potential instead of having strange edge features, and that could potentially interfere with your folding simulations. And then for the L-BFGS folding simulations, we parameterize the RNA structure using the backbone eta and theta pseudo-torsion angles. And then we just follow the standard L-BFGS updates. So in this case, we assume all bond lengths and bond angles are ideal, and that we can just recapitulate the structure based only on the pseudo-torsion angles. And so if you want to calculate the update for your eta pseudo-torsion angle at step k+1, it would be based on the eta torsion at step k plus alpha k here, and dk. And so alpha k here is just the step size, and it's basically determining how big of a step you're going to make in the direction that's determined by dk, where dk just relies on the gradient of your energy function with respect to the eta and theta pseudo-torsion angles. So I think it's a fairly standard implementation of the L-BFGS folding simulations. And so, of course, the nice thing about these is that they are very fast, at least for the folding procedures, where we can typically fold about 100 nucleotide RNA in 1 minute. So if you look at this case, you can see that the global structure of the RNA is determined pretty rapidly within maybe the first few hundred steps. And then once you get towards the end of the simulation, you have more minute changes in your side chain packing and trying to resolve the structure at a much more local level. But as I mentioned, it's significantly faster than, for example, the molecular dynamics based methods, or the Monte Carlo based methods that are typically used in the folding procedures. So now I think I'll cover the results. Yeah, so here's just a table from the CASP 15 assessment looking at the results on the targets compared to all the groups that participated. And one of the things that was the takeaway from this was that the physical energy or the physics-based methods seem to significantly outperform the deep learning-based methods, which, you know, I think it's probably because of overtraining. So I know in the original data set, it was set at 80% sequence identity cutoff, which of course now we discover is certainly not stringent enough for an accurate assessment of what your performance should be. So what we see here is that basically all of the top groups are those based on the physics-based methods, which with input, I think, from the experts in the field on the RNA structure prediction, which is another interesting, I guess, difference in protein design is typically there isn't too much extensive intervention. Typically it's run in a fully automatic method, where there's not really too much time, I guess, to perfectly perfect all of your targets. And it seems like in RNA that it's actually very helpful, the insight from the experimental experts and experts in the RNA structure prediction. So what we see, actually, we could.
Sorry, Robin, can we go just back to the slide? And maybe you just said it, but can I ask again? So in your case, the procedure was fully automated. Yes, you didn't
Yeah,
play.
sorry, I should say that.
Okay, so that's
So the procedure was fully automated, based on from start to finish, essentially, from the model generating the network restraints to the model selection. So that's what we really wanted to do, is to have a method that didn't rely on humans to run it. So I think
And here there is DF_RNA, and is it, because to me at least it's a little bit confusing, because at the webpage of Zhang, and there are like two papers. One of the methods is like DeepFoldRNA, another one is DR.
Oh yeah. So
can you explain this a little bit
yeah.
here?
Yeah, so I think we publish the RDP. The DeepFoldRNA we published, we just put it on bioRxiv first, and then later we worked on RDP, which is the difference between the two methods is that RDP uses the end-to-end deep learning training, as well as single sequences. So there's I'm presenting the DeepFoldRNA, not the RDP.
Okay, but here so the method here, the F_RNA, is your method?
Yes.
Okay. Thank you. Just to make it clear.
Yeah, absolutely.
Okay, great.
Yeah. So it's interesting that just looking at a subset of the different targets, because it seemed this year that, or at the last CASP, that there were essentially, I guess, two types of targets, where they had, there were natural RNA targets, and then there were these large synthetic aptamers and six-way junction targets. So first, I want to just compare the performance on the subset of natural targets that we submitted models for. So the performance of DeepFoldRNA is shown in the red star, and this is based on the best of the top 5 models submitted by each group. So on the left-hand side, you have the TM score, and on the right hand, you have the RMSD. So if you look at the TM score and the RMSD, on the natural targets, it seems that the deep learning methods are quite competitive with the physics-based methods. And I don't think that any of the structures were available beforehand. And I think several of them didn't have natural structural analogs yet in the PDB. So it seems that the methods generalize at least reasonably well on the natural targets. And if we look at the, let me present. So I'll just talk about 2 of the case studies for DeepFoldRNA here. So the first one is from R116, which is the cloverleaf RNA. And actually, this was the target in the CASP test set that had that we were able to generate an MSA with the highest NAF value, which is generally used as an indicator of the reliability of your MSA and its quality. And we have the predicted structure on top, or the native structure on top, and the predicted structure on bottom. And we can see that, so if you look at the first portion of the structure here, it seems to match quite well with the native structure. And then it looks like there's some twist or some deviation at this point in the structure that causes there to be a high RMSD. So the RMSD is around 15 angstroms, but the TM score is relatively good because overall the global fold for this case was able to be successfully recapitulated. But because of this weird twist going on in the predicted structure, the RMSD was significantly higher. And the second example is actually from the targets from CASP that had the lowest net value, meaning supposedly the least reliable MSA. So we have the native structure on the left and the predicted structure by DeepFoldRNA on the right. And this is for the SARS-CoV-2 SL5. And so overall, if you look at the structures, we obtained an average or a TM score of 0.536, and then an RMSD of 6.88 angstroms, which was the best among all submitted models. And it seems like the major region that we missed here was we missed this long-range interaction between the, I believe it's the SL5A region of the structure, where it comes in and forms contact with the remainder of the RNA chain. And it's interesting, as I alluded to, that this was the target that had the lowest net value. So it had a net value of about 0.292, which is quite low. And you can see from the sequence profile that essentially all of the sequences that we picked up in the majority of the positions were almost identical. And another interesting thing is that if you look at less conserved regions, for example, from 113 to about 118, these are residues that fall within this, I guess, this little pocket here. And what we could see from our submitted model is that that would certainly the lowest confidence region for us. So maybe there's some flexibility or some uncertainty in the model prediction due to the sequence profile of that position. But overall, I think it's an encouraging result because we're able to produce a fairly reasonable structure with starting from an MSA that may not be so reasonable. So here's just a look at the rankings from the groups based on the left hand here, you have the sum of the z-score greater than 0 for the TM score. And on the right hand, you have the sum of the z-score greater than 0 for the RMSD. And these are for all the groups on the natural targets. And we still see that, for example, in the TM score, we have the top 3 methods are the physics-based methods, but we're much closer overall than we are on when considering the synthetic structures. And we're even closer if we consider the RMSD, the z-scores of the RMSD. And not just our model, but I think a lot of the deep learning models like AlterFold and AI Chemi RNA and the AlterFold server, they're all right up there with these top physics-based methods that were guided by the human experts. So I think at least it's a somewhat promising solution for the fully automated structure prediction that we're able to get fairly close to those state-of-the-art from the physics and the experts based folding methods. Okay, so now here's where it all goes wrong, is on the synthetic targets. So if you look at the results on the synthetic targets, we're we, again, the DeepFoldRNA results are in red stars in terms of the TM score and the RMSD. And we can see that we're not doing very well, especially not on the RMSD. The RMSDs are generally very high. And of course, the physics-based methods it completely outperformed us. And I think a lot of the deep learning based methods failed pretty catastrophically. So, but I think that's, one of the things is that here's just a depiction of the three synthetic structures, is that all of these large synthetic RNA structures, they had no homologous sequence information. So meaning, at least when I ran the MSAs, I wasn't able to pick up any native sequence analogs. So essentially we're running single sequence based predictions. And also I think that the folds themselves may not be indicative of what you would see in a natural RNA. And of course, the methods were all trained on natural distance distributions from native structures. So I don't think it's too surprising that they would not perform well on these very non-homologous targets. I think that's probably been established for a while in protein structure prediction, where generally the fewer or the less robust your MSA is, the worse your results will be. And especially for the synthetic designs, or maybe for the de novo designs in protein, where I think it can be quite difficult to predict those structures at times. So let me see. It's until 12, right?
Yep.
Okay, let me finish up real quick. So lastly, I'll try to run through this. To try to further test the performance on the natural RNA structures, we collected a set of 96 RNAs that were released after we collected our training data set with lengths between 30 to 300 nucleotides. And we eliminated structures with homology to any previously solved RNAs using a BLAST eValue threshold of 10. And so we don't have many structures here to compare with, but we have about 7 structures, and 3 of those structures possess novel folds where the X marks the novel fold. And then we just compared with the fully automated version of SimRNA against DeepFoldRNA. And so overall, on these native structures with low homology to the training data set, we're able to achieve, I think, fairly robust results. And I can just show maybe one example, and then we can take questions. So here's an example from the, oh, sorry, my screen. Here's an example from the dengue virus RNA promoter, which was one of the novel folds. And the SimRNA model is shown on top, and the DeepFoldRNA model is shown on the bottom here. And what we can see is that, again, this target had a very low net value of about 0.32. So we only had 3 sequences in the multiple sequence alignments, and yet we're still able to get a realistic prediction, or at least a pretty good prediction, with an RMSD of about 3.7 angstroms. And the interesting thing about this is that the structure is from an NMR ensemble. So I think there's some flexibility in this molecule inherently. And when the authors, after the authors solved the NMR ensemble, they looked at the SACS envelope as a way of validating their results. And it looks like the top of the structure here from the NMR ensemble sort of peaks outside of that SACS envelope. And we can see that for our predicted structure, it sort of resembles the same pattern in that region, where most of the deviation between the two structures is in that maybe somewhat less confident region in the NMR ensemble. So let me see. I'll skip this for the sake of going to questions. So yeah, so just in conclusion, starting from sequence alone, we can robustly fold the natural RNA sequences, it seems, based on the blind tests in CASP, using a fully automated approach. But of course, the performance on the synthetic RNAs was very bad. So, but the some of the ways that we may be able to improve that we've been working on recently is incorporating these pre-trained language models that we can leverage the unlabeled sequence data. So in proteins, we have methods like ESM-fold and others that have shown that the embeddings learned by the pre-trained language models, although they weren't specifically trained on the structure prediction, that information can be inherently captured by those models. So I think we've been doing it now, and I think the results are pretty promising. And what we also like to do is, of course, extend this to the protein RNA and the RNA interactions. And we can do this pretty easily through the end-to-end modeling as well. So previously, what I presented was based on the restraint-based modeling, but it's fairly straightforward to extend that to the end-to-end structure prediction, which is very convenient for complex structure prediction in particular. Okay, so yeah, that's all I have. Anybody has any questions?
Okay, thank you again for a wonderful talk. And do we have any questions from the audience? I don't see.
I have a question.
There is one.
Okay. I don't know if my mic is working.
Go ahead. But yeah, speak loud.
I have a question about the data you use from PTV.
Yep.
You use only the standalone RNA, or you are mixing the RNA that interact with proteins and so on?
I was a little, I couldn't quite hear what you said.
I think the question was, did you use only RNA only structures, or also protein, like protein RNA?
Yes, that's right.
Yeah. So I used protein RNA structures as well. So I didn't consider RNA only structures. I mean, I included both. So.
Okay, Janusz.
Hi, great talk and great method. I have a question, but I would like to start with a comment. So it's great that your method performs so well on structures homologous to the other ones. And I'm not surprised that you're much better than simRNA, because for simRNA, essentially a structure that is well known, like a tRNA, is as hard as something that is like a completely new fold. It does not consider any information about homology to existing structures, or any similarity to existing structures, and it does not use any alignment information explicitly. So I'm not surprised that you beat simRNA in such cases. My question is related to the use of the alignment. How well does your method perform if you don't use alignment, or if if you have a single structure or a single sequence that shows no similarity in databases to any other RNAs? How well does your method perform? Because this is the true situation for most of the RNAs, where you have only one sequence, or just a few which are super similar to each other, like 4 sequences that are 90% or 95% identical. So what is the performance there?
Yeah, so that's exactly this case. So R1149.
No, sorry, no. SARS SL5 region has multiple sequences that are similar to it. So this is a bad example, but please continue.
So yeah, we calculate the similarity based on the NEF value, which is the number of effective sequences, which takes into account those sequences that are highly redundant. it tries to remove the influences from, for example, you have 20 sequences, but they're all very redundant, meaning there's greater than 80% sequence identity between them. So I think it goes to exactly what you said, because we were able to collect about 30 sequences, and out of the 30 sequences, I think 25 or so had greater than 80% sequence identity. So I think that this is exactly the case, because as you can see from the NF value in the MSA, which measures the number of effective sequences, that's it's quite low. So meaning that all of the sequences it picks up are highly similar.
Yeah, so I'm sorry. I think you misunderstood me a little bit. For me, this case is like, well, not great, but already good sequence alignment. And having like more than 3 or 4 sequences with 80% identity is already useful information. I'm talking about cases where you have just a few sequences, like literally 5 tops, and and almost identical, with almost no gaps, almost everything identical, like 90% identity, or even just one sequence. So I understand. I appreciate your response, but this does not really address this type of difficulty that I'm asking about. And I'm not criticizing. I'm just asking, how well does your method perform in cases where you have like almost no alignment?
Yeah. So I mean, in this case, we have 2 sequences. Of course, they're not 100% identical, but the alignment is very sparse. So the net value, I think, is 0.32. So we have the query sequence, which I don't know if you can really count. That's just the individual query sequence. And then you have 2 natural homologs. So I don't think that's, and also for this target, it's a new fold. So there was no other previously solved RNA structure with the same fold. So I think it sort of answers both of the questions, where can we generalize to new folds? And can we effectively predict those targets with very bad sequence profiles or bad multiple sequence alignments? And I think that we can probably do it. How often we can do it, I'm not sure yet, because it of course relies on there being new folds released. So, but I think that one of the ways that you could answer that is that when you do the training, is you could just exclude all of the training samples with, for example, any structural similarity, and then you have a completely non-redundant structured data set. So I think there are ways to answer that, but based on the presented results here, I think that we're still able to at least obtain somewhat reasonable predictions.
No, certainly, and your predictions look very good. This, I don't know the structure. It looks kind of kink turn-ish to me, but I need to take a look at it. I don't remember what are the exact interactions. Thank you very much, and good luck. And we'll definitely use your method in our pipelines. Thanks.
Yeah. Okay, so maybe I will read Regiu's questions, because it kind of groups with question of Janusz. So RNA Puzzles has had several natural RNAs recently without homology to previous solved structures. Have you tried DeepFoldRNA on some of these post-CASP blind tests?
We didn't participate in blind tests, but I have run it on the structures. So yeah, I mean, I think it does about as well as what we saw on the blind test for CASP, the RNA. About as well as what we expected from our results on the natural targets in CASP 15. So I don't have the data here, but it's a similar, I think, a similar trend where for the natural RNAs, we're certainly able to generate much more reasonable models than for the synthetic RNA. Okay,
Regiu says thanks. Sorry, Conan, I could see your question in question and answers, because now he wrote it again. So I will ask Conan's questions. How much fraction of native RNA structures from PDB as training data belong to ribosome structures? Do you know
Yeah, of
roughly?
course. So a huge majority of the will belong to ribosome or tRNA because the diversity in the number of folds is very limited in the PDB training set. But I guess all the test results presented here, none of them should be tRNA or very well represented structures from CASP15. I think they all should be fairly novel.
And there is one more. Have you considered probing data, shape, DMS as an input in your model, or even SAX data as you showed?
Yeah. So absolutely. So I think it's fairly straightforward to incorporate that. So right here we have our predicted secondary structure. You could essentially just replace the predicted secondary structure with whatever pairwise probing data you get or contacts from maybe shape to get your secondary structure. So I think there's room to incorporate experimental information into the method.
Okay, thank you. And there is one from Craig. What does the single sequence prediction network predict on randomly generated sequences? Can you tell a real structured RNA from random sequences? Simple 2D folding struggles to do that.
So what does it predict if you give it just a completely randomized sequence?
Yeah, so
Yeah, that's a good question. So I haven't done it yet, so I wouldn't have a concrete answer for that. But I'm guessing if the sequence was just completely randomized, then your distance maps would also be. I think they might just be random noise, or I'm not sure. I would have to see.
Yeah, because I think the
Yeah.
point of the question is that it could. Oh, okay, Craig.
Oh, yeah. Thanks, Robin. Great talk. The reason why is when you run that through 2D structure prediction, it finds helices all over the place.
Yeah.
And
Yeah.
so what will the 3D do? I mean, if it's a quick run to rule out that some RNA is a fully structured 3D RNA, that would be nice to have.
Yeah. So what I've seen that what happens when the sequence is maybe somewhat low complexity or just like repeated adenosine, that it just essentially is unfolded coil will be the output. So, but I haven't really done a full, that's just based on my anecdotal experience from what I've seen when I run it.
Sure. And similarly, you could take an alignment and shuffle the columns, and then you'd have covariation in the columns. Anyway, might be interesting.
Yeah, absolutely.
But not so pressing.
Yeah.
Okay, so maybe the last one. So Conan asked again, which steps or modules of the DeepFoldRNA network pipeline affects most of the final prediction quality?
Yeah, so essentially the triangular attention for the processing of the pair embedding, because at the end of the prediction, you're using this pair embedding to get your distance and your orientation maps directly. So I found that when I leave out the triangular attention, that the network is much worse.
And yeah, and I guess as an input also alignments, but yeah, they are not part of the network itself. Okay, so I don't see any further questions. I think it was very informative. So I hope soon we'll upload the video. And thank you, Robin, again. I think was really good talk. And yeah, Craig, Janusz also say thank you.